because syllabus is used. Okay.
Okay.
So the possibility is there that you should save your energy and time.
Yeah.
So, and always keep on discussing. Wherever you are feeling doubt, don't hesitate, because if you are hesitating with the doctor and teachers, then you cannot rectify your problems.
Yes, sure, sir.
That's a big thing. And
Oh,
whatever the section C is there, that is experimental part, right? That is
yeah.
applied part, you can say.
Yeah. Yes,
I
sir.
will I will make I will explain you in the detail. First, what is
Yeah.
the syllabus? Because that's a very important, and then we will go through it.
Okay.
So,
Okay.
because before cracking the exam, before playing a game, you must understand the rule. That is the policy, right? And if you understand the rule, then you can crack it. No one can stop you. So let me explain you the syllabus, how to work with that. That is very important.
Okay.
Just a minute. Before many students join, I said. Okay, now you can see the screen, basic genetics.
Yeah. Yes, sir.
Okay, let me explain the syllabus first, and then strategy. Our student is adopting mostly. This is syllabus. Okay. So syllabus is divided into basically 3 parts, and whatever the time you are getting almost 3 hours,
Yes.
and the maximum marks is 200, right? So time is very less. You can see
Yes.
the part A, part B, and part C. And this is the most scoring part, right? So in part A, there are how many questions? There are total 20 questions.
20.
Okay. 20 questions. You have to attempt only 15 questions.
15 questions.
Okay. So this is
Yeah.
a 10th question, 10th math. 10th maths. Okay, research aptitude, little bit, and reasoning, quantitative reasoning is there. For example, if you are going left, then what will be the next? Right? So they can give you just thought type questions, and you have to apply your brain, you have to analyze, you have to visualize, and then you can solve it. So you can
Oh,
practice the 10th class maths, and basically time, watch, clock, you know, from
mm.
clock, from time, from speed, velocity. No, you should note down, no? Mensuration, little bit of geometry, little bit, very little bit, but mensuration is major part. Speed, velocity, at least 2 questions are there. Mensuration, 1 or 2 questions. Clock, 1 or 2 questions. Time, you know, time is related to the clock itself. Okay,
Yes.
so these are few portion, and analogy is there, logical reasoning is there, age related question is there. You know?
Yes.
So these are few questions you can find on the Google or any kind of 10th maths book. So you can
Yeah.
practice that one,
Yes. Yeah.
and it's not tough. And this decides actually the GRL. This portion decides. So you'll be surprised to know, because this is carrying 30 marks. And if you are, at least you are gaining the 15 marks, it's a big lead,
Yes.
because nowadays you can see the score is going. Our 2 students has got the score card 98.9 percentile. So you can see such a huge scorecard.
Yes. Yeah. Yes.
Means you have to keep calm, you have to have patience. Our student first time she given the exam, and she scored 85 percentage, and now
Okay.
she is doing the internship from Panacea Biotech.
Wow.
Right. So I think she will join us today,
Okay.
because she's working late night, so often she becomes late.
Okay. Okay.
That she will join. She may join. So many students are studying in proper way, because we are not coaching, we are kind of guidance platform. We are charging very little because student then attending the focus, right?
Oh,
And
yes.
not paying, if it's a free, then mostly students are becoming the casual. Okay, it is sometime, if you are paying something, then you are very careful.
Yes.
That
They will take
gives
that
a lot
they
of
are
success.
paying and they should keep attention more.
Right. And we are charging only 750 rupees per month.
Okay.
That's only for internet charges.
Okay.
Right, because 1 GB data every day we are, more than 1 GB, because it's a heavy stream platform.
Yes. Yeah. Yes.
So part B is almost 50 MCQs you are getting, 50 questions you are getting, and out of that you can select any
Yes.
35 questions, and each questions are of 2 marks. So
Yes.
35 into 2, it will be almost 70,
70.
right? So
Yes.
70 marks. So it's almost my students are crossing the more than 30 questions.
Wow.
This question that is called
Mm.
question. And why? Because it needs a deep understanding. MCQs are always, MCQs choice can be anything. You have no
Mm.
freedom to write in your own way. So you have to be very cautious when you are studying, when you are studying the question, because reading a question carefully is very, very important.
Okay.
Very carefully, because sometimes you see a very common word, but its meaning is
Mm.
totally different in the options. They
Yes.
can ask you the different way. So because CSI are never going to repeat any questions. Always the scientists are there and they are going to frame the question in a very new way. So
Oh,
uh, we are practicing our module, our unit-wise test is there on the Saturday or Sunday, right?
mm.
So mostly Saturday students are studying in their own way, and on the Sunday evening we are allotting a questions and
Okay.
CQs, right? Practice test over the app. There is a app. Have you downloaded the app?
Yeah.
There is a Merit Life Sciences app.
Yes.
So if you are enrolling, then we are allotting a batch, right? In the batch
Batches.
22, you will be getting the all type of study material, questions, notification, class notification, right? For example, you
Okay.
got the notification from the app, I think,
Yeah.
right? So study
Yes.
material and then test. This test you have to ensure that you are getting not less than 80% in each test. It must be
Okay.
80%. And you are allowed to do the mistakes not more than 5 questions,
Okay.
because there is a negative marking in each questions. This is the third part, and it requires at least 1 hours. Right? You cannot exceed that hours, because part B also requires 1 hour.
1 hour.
Okay. Ha. Right. Hello, Luni. I'm audible to you.
Yeah, your voice is
Can you
breaking
hear
now.
that?
Your voice is
Okay.
breaking.
I'm just now it's okay.
Yeah.
Check
Okay.
that. My voice is clear now.
Yeah, it's
Because
clear.
sometimes internet speed is down,
Yeah,
so
it's
streaming video kind of hmm, that's costly. So you requires at least 1 hour for part C, and this is the most scoring part.
yeah.
For most of the students, we are advising to start with section C, and you are getting the 75 questions. You have 50 questions to leave, right? You have to attempt only 25 questions out of that, and each carrying the 4 marks.
Yes.
Right. So these are going to tell the scientific concept or application of the scientific concept. So this part we can say totally
Experiment.
experimental, means condition based questions.
Yeah.
So this is totally different from your previous studies because we
Mm.
have to understand the concept here. You cannot mug up the concept. You have to understand the concept. For
Yeah.
example, if I'm asking, what is the penetrance in codominance? What is the penetrance in codominance blood group, for example, right? Blood group is the best example of codominance. Option A is 25%, option B is 50%, option C is 75%, option D is 100%. So try to answer this.
I guess it's 50%.
Why not?
Hello.
Please.
Sir,
What
I
is the
guess
reason of being it 50%?
both A and B alleles are there, no, sir.
No, any specific reason for being 50% or just you have guessed it?
Not, I cannot explain, but I guess it's 50%.
Okay, the answer is correct. And the reason is what? You must know the concept of penetrance. Then you will be sure. Yes, I
Okay.
marked it correctly. You are not going to confuse. Because penetrance means if suppose there are two alleles,
Mm.
dominant and recessive,
Recessive.
right? So
Yeah.
if dominant allele A is there, then recessive allele is going to be suppressed.
Suppress.
In that case, you can say there is a 100% penetrance of dominant gene,
Okay. Yeah.
right? There is a
Yeah.
100% penetrance.
Yes.
And suppose they are co-dominant, for example, in the blood group AB,
Immunoglobulin A, immunoglobulin B.
right? So in
Yeah.
the case of uh, blood group AB, you can say 50% contribution is from A and 50% is contribution from B in the term of penetrance. So
Yeah.
overall penetrance will be the 50%, right?
Okay.
In the case of suppose incomplete dominance, what you will say? The percentage penetrance will change, right? Penetrance will get changed. So
Okay.
obviously the answer will be same, right? Answer will be
Yeah.
same. But in case of albino, which is a kind of lethal gene, when the recessive character are there, recessive gene are there, then it's going to die. Then what you will say? What will be the percentage of penetrance in this case? So this kind of question you are getting in the part C.
Okay.
Condition based means you are not going to find anything in the books.
Mm.
Always you have to think when
Like,
you are in the live classes. That is the
yeah.
benefit of live classes. Means you can discuss, you can express your thoughts, you can
Okay.
make a argument, right?
Yes.
Daily we are allotting some questions and you have to solve it. And then you have to return back next day.
Okay.
Right?
Yes.
That's the concept of practice makes man perfect. And we have to, since we are teacher, we know we check always the capability, means IQ level. right?
Okay. Yeah.
Percent,
Yes.
percent expression of students.
Mm.
Okay, so that's accordingly we are modifying our lecture
Okay.
because we know everything, but students' level decides
Yeah.
what should be the level of the lecture, and slowly we are going to deliver in the advanced mode,
Okay.
right?
I have to share
So
one thing, sir.
yes, sure, welcome.
I qualified in AP RCET, for Andhra Pradesh
Yes,
Research
yes, okay,
Entrance Test. I got third rank for full time PhD.
okay, that's good.
Actually, I had counseling, but I don't know whether I will get funding or not.
Funding is not sure when you are going through university test, but this is reserved for only 10% people.
Okay.
So you
So
might be lucky
after
and
joining,
get,
I wish to crack CSIR. It's my wish.
yes, देखो, uh, CSR is a concept, right?
Yeah.
If
Yeah.
you have a complete concept in your mind, you can crack anywhere, right?
Oh,
And if you are qualifying GRF, then any university can take you as an internship or GRF, sorry, as a PhD student, right? There is no issue. Why? Because if they are asking, right?
mm, yeah.
Suppose there is a war, atomic war, right?
Yeah.
So, what will be the effect on human being? So,
Mm,
uh, from the 10th point of view, you can say that all the animal will be affected, in the plants will be affected. But they can ask you the different question: how will you save
yeah.
yourself, right?
Mm,
Or what kind of plants are there? What kind of flavonoids are there? In the depth study, they can ask you. They can
mm,
save you.
mm.
Do you have any idea? So, if you are a biologist, then you must know that there is a osimum tulsi, very popular
Yes.
plant, right?
Yeah.
And then cow dung. These are shown to prevent any kind of radiation. So in the
Mm,
old times, we have seen that mostly the homes were not made up of bricks. They were made
mm.
up of soil and cow dung laid by the cow dung, okay, in outer wall. There
Okay.
is
Yeah.
no kind of wall putty and all that, right?
Yeah. Yes. Yes.
So that was kind of concept. If any kind of huge radiation are there, so that can protect. And if you are taking the osmum tea or kind of eating, and suppose there was a corona, right? COVID. And in the COVID-19, who were aware, they were taking, they were hugely using that one. So that was directly preventing, that has a component,
Mm.
uh, assume is a component that prevents TNF alpha beta activation.
Okay.
Right. So that is directly involved in the inflammation, fever, any kind of virus attack is there. So they
Oh,
are they are released hugely in the body, and they are responsible for huge fever and all that symptoms. All right. So
yes, sir.
practical based aspect they can discuss, right?
Okay.
Practical based discussion maybe there.
Yes.
So one more student has joined from Assam,
Okay.
right? And Lanu from Nagaland. Just we have discussed. And Pipanchi,
Good evening, sir.
good evening. How are you, sir? So you are from Assam, na Pipanchi? I think I
Yes,
got
sir.
it. I am just checking my memory. Yes. So today Brahma Kalyani has joined from Telangana, Hyderabad. So we are
Hello,
discussing
Kalyani.
several parts.
Hi.
So she wants to qualify the CSIR, but little bit facing the problem. She already has qualified the Andhra Pradesh research exams. So we are discussing that there are three portions, and basically the portion C is very uh, scoring. That you have to understand. That's we are mostly discussing during the discussions, because if you're teaching the same thing written in the book and if you're not discussing the practical aspects, it's of no use. So we have discussed some few lectures from last 2 days, the dominance, aggregation, dependence, assortment. Yesterday we have discussed about the alleles, multiple alleles, pseudo allele, complementary syndrome. So today we are going to discuss this concept: codominance, incomplete dominance, gene interaction, pleiotropy, genomic imprinting, penetrance and expressivity, phenocopy, linkage, crossing over, sex linkage, and sex influence factor. So I think this is a very long topic. So we are not going to discuss this one today. Right? We are going to discuss this one only. Right?
Okay.
So codominance already discussed. Best example is the blood groups. Incomplete dominance, pink flower of a rose. We have discussed yesterday. That is the result of incomplete gene interaction. Today we are going to discuss about the phenocopies and some pleiotropy. Genomic imprinting is a very wide topic. This is related to the genome silencing. Right? This is a kind of genome silencing. Are you aware of the genome silencing? What is it?
Functionality of gene is
Affected.
affected.
How that expression is affected. I can give you example. For example, you can see the egg, right?
Yes.
Right, fertilized egg. You can see. So there are several genes which are silent. Okay. Only the first anterior gene is going to express, then posterior gene is going to express, and after ensuring their expression that they have expressed perfectly, then middle gene is going to express. So this is the best example of genome imprinting, right? So it has been found that totally gene are there. Gene means mRNA. Because mRNA is going to form the proteins, right? So if mRNA is methylated, then what will happen? And if heavy methylated, then what will happen? Any guess?
It won't. That means translate into protein.
Right, because there is a blocker. They are acting as a blocker. That makes the genome silence, right? And after removal of this methane, the gene can express. So if there is a completely blocking of a gene can happen if there is a heavily methylated.
Okay.
Right. For example, you can see the eukaryotes after the formation of mRNA in the nucleus, they are coming out in the cytoplasm. They are coming out in the cytoplasm. So what is happening? Immediately they are get methylated, right? Immediately they are get methylated. So this question was asked in the CSIR. When there is a eukaryotic cell or our cell, and they are infected by the viral DNA. So there is a fast DNA, right? And they are inserting some DNA inside the eukaryotic cell. So how our cell, our eukaryotic cell, our bacterial cell, they are going to defend themselves. How they can defend themselves? Means they are asking question in different way. They are asking question how the cell going to identify between our own mRNA and viral mRNA. Especially they are not cutting our own mRNA rather than viral mRNA is going to be attacked. And they are going to be fragmented into the pieces. So what is the reason? Can you guess?
Our
Simple reason
mRNA
is,
is methylated.
yes, they are immediately get methylated before they are coming out in the cytoplasm. Then only they are protected, right? And viral mRNA are not going to be methylated because membrane, at the membrane, if they are methylated, they cannot enter inside the cell. Right?
Okay.
So they are demethylated mRNA, viral mRNA, demethylated. Right. So that's why the whatever the enzyme restriction enzyme is there. Right. They are going to they are first identify demethylated mRNA or DNA.
Okay.
And then they are attacking, they're making them fragments. And that's why this strategy used to make a safeguard of our own mRNA.
mRNA.
So that is related to the gene silencing. And if you are saying the gene silencing in the term of genome packaging, so you might be knowing nucleosome model.
Yeah.
So this is a H1 histone are there. Different histones are there. H1, S2A, S2B, S3, and then one H4, right? So you will see these nucleosome also are associated with the methylation pattern, right? And once in the CSIR, it was asked that what is the force involved in association or strong binding of DNA with histone. The option is covalent bond. B is hydrogen bond. C is the ionic bond. B is not. Got the question?
Yeah.
They're asking about the force that is involved in strong binding of DNA with histones. What would be your answer? So you should understand that these histones are associated with the what? Some unique amino acid. Can you guess which amino acids are there?
Hydrogen bonding.
No, not hydrogen bonding.
Covalent bonding. CH3, CH3.
No. Covalent bonding, if covalent bonding is there, then certainly they are not going to open at the time of replication.
Okay. Yeah. What is this third option, sir? I am not getting. Nick bond.
Actually, DNA is negative charge because of phosphate, right? Because of phosphate, DNA having the negative charge, and because of positive amino acid, basic amino acid, this histone is having the positive charge. So there will be electrostatic or ionic bonding between them.
Okay. Achha.
So you got the idea. It's not never going to be discussed in the books.
Okay. Yeah.
So yeah. Now you can identify that what you have to do during the discussion, right? So if I'm telling there is a negative charge in DNA, then you have to ask me why this is negative charge. So then I am telling phosphate is there, right? And if I'm telling about the nucleosome, then you have to see in more detail. There are 4 types of histones, 5 types of histones. Then you have to go into the much more detail, right? Is the histone is associated, how many amino acids are there, which amino acids are there, what charges are there. You have to think in your terms. There is no one going to tell you. then you will find the answer. Because
Okay.
everything has been researched. Right? So you have to make a query with yourself, with me. That is the benefit of study in the live classes.
Okay.
Vanchi, is it comfortable today?
Yes, sir.
Okay. So this is very good discussion today. Now I think you got the idea that in what way we should prepare the handle the syllabus, right? And what would be the strategy to for the perfect learning. So whenever I'm discussing something, you must ask the question because this is kind of discussion platform, and that then only it can be useful. This is my brief bio. You can see I'm guiding one PhD student currently. Four patents are there, two textbooks are there. You can Google them. And I have almost 20 years of teaching experience in different reputed universities of India. I'm from Banaras Hindu University. In biotechnology department, I did my PhD and over 3 novel things. I have the 4 patents. I guided at least 200 MTech and MSc thesis. And I have published more than 60 research papers. Right?
Yes.
So this is my brief bio. And from last 3 years, I'm a passionate teacher. So I'm teaching to my CSR and GRF because there was more incident when I was guiding some students. They were not able to qualify even after paying a huge amount of money to the different coachings. So I got research over that, and then I find that all they are just giving the information. There is no conversion of information into knowledge. Right? Information is useful when you are analyzing it, when you are applying it. Then it's converted into the knowledge. Agree?
Agreed, sir.
Okay. So our today's goal is understanding the pleiotropy, genomic imprinting. Already we have discussed a little bit. Penetrance, expressivity. We have discussed a little bit. Phenocopies. Do you know the phenocopies? Very interesting. Have you any idea of the phenocopies?
Looking similar.
No. Exactly. Suppose there is a mouse. चलो ये mouse मेरा बन गया. So you are raising them at 30 degrees centigrade, and if you are raising them at 40 degrees centigrade, you can find a change
Okay.
over the phenotypic character of both. So that identifying the reason why there is a change. Frog. Right. So I'm not a good artist, but mostly whatever the diagram I'm using, mostly I'm making themselves. So this is the effect of exactly what you can say a term. Phenocopy is the effect of environment over the expression, gene expression. They are changing the gene expression, and that results in the changed phenotypic character. Changed phenotypic character.
Okay.
All are requested to please make a notes, okay, and try to attend the class regular. Be interactive, right? When you are interactive, then it's more useful because you are mature students, you are grown up students, right? You are not kids.
Yeah.
I'm lecturing and you are listening silently. Then you cannot enjoy the sessions because science is to enjoy the sessions.
Yes.
Okay. So if I'm talking about the phenocopies, there are a lot of example. I can show you. I am going to discuss this one. This is phenocopies.
Okay.
This is the best example of phenocopy. You can see, if you want, suppose you are in this area and you are selling the animals. Especially the rabbits one at your home. Then just you have to keep in mind that temperature can affect their expression differently. Okay. Right. So at 20 degrees centigrade, what is happening? They are also expressing black, blackness over the ears, mouth, legs. But
Like
if you are changing the temperature, raising the temperature at 30 degrees centigrade, you can
everything.
see the everything white. Why it's happening? Why it's
Due to
happening? Because of effect of allele expression, which is temperature dependent.
Dependent.
Because it's temperature dependent. So phenocopy means effect of environmental change, and that's going to inhibit, either they are going to inhibit the expression of some genes, right, or they can make more expression of some genes. So both the cases are possible. Either they can inhibit, either they can making the gene more expressive. So that genetically inherited trait at certain fixed environmental condition, that's scientifically called as the phenocopies, right? So that's only because of change in the environment. So it was concluded that always allele not going to show the same effect, right? Sometimes the effect can be changed if you are going to change the environmental condition. So why, how the environmental condition is going to act? Actually, allele supposed to express at certain environmental condition. But if the environmental condition is not favorable, right? So that gene expression can be inhibited, right? So exactly the allele is related, surely allele is related to phenotype, but not always. Because in centigrade, the black alleles are going to express here, right? Black alleles that is responsible for the black coloration over the ear, mouth, and leg, they are going to be expressed, right? But at higher temperature, so for the new students, we have just started discussion over the extension of Mendelian genetics. So today we are going to discuss following topics: pleiotropy. What is the pleiotropy? Anyone?
One gene have a more than one character, sir.
dominance.
Right, right. When it is possible, when more than one gene is going to control one trait, when it is possible.
Sir, more than one gene will control one trait, right, sir?
Right. This
तो
is
ये
called as the pleiotropy. When this is possible.
एपिस्टेसिस में होता है.
Linked
ये तभी होता है
genes.
जब एक ही क्रोमोसोम पे सारे जीन लगे.
हाँ सर.
All the gene present in the cis position. यानी same chromosome पे होंगे, then only they can control the more than one trait, right? Otherwise, what will happen? If they are on the different gene, they cannot control the same trait. So many cases have been reported. Yesterday we have discussed it in detail, right? That more than one allele are there that are controlling the one traits. So a little bit of recap. Yesterday we have discussed about the loss of function. So we discussed that loss of function is operative only in the recessive state, right? And when the gain of function is going to act, when there is a wild type allele are there. Right? Wild type allele is going to give this opportunity, that is gain of function character in the mutation part. And if the recessive gene is there, always they show the loss of function, right? So formation of, for example, formation of a toxic product that can kill the cell is not possible in the recessive state. That is possible only in the dominant mutation case, right? You got the idea? Suppose there is a toxic product formation. So if dominant mutation is there, dominant gene is there, then only they can be produced, right? They can be functional, right? And they can kill the gene. Otherwise, in the recessive condition, this toxic product is not, expression is not possible. So mostly loss of function is there, means their expression is going to be lost. Gain of function, हमने कल बताया था. Suppose this is yellow color expression, and if there is a gain of functions, might be their color is going to be changed in the animals. Suppose this is a fur color in an animal, and because of gain of function, what can happen? Their color might be changed. Okay, you got the idea? Any
Yes.
problem?
Yes, sir.
So this is very simple concept we are discussing already. You might be knowing many things. Autosomal recessive. This is mostly asked question, very confusing questions. So you must learn the examples first. What are the example of? And if you can remember in 2020, 2019, the question was repeated and asked in the different aspects. All right?
Yeah.
CFTR. So CFTR, widely known as cystic fibrosis transmembrane conductance regulator gene. And this is occurring over the chromosome number 7. Right? So if they are going to be dysfunctions, they can cause the bronchitis. Right? So this is mostly occurring sometime in the children, sometime in the adults. And this is only because of AR, autosomal recessive disorder. Means if the two allele carry the same mutation, the individual is homozygote. Homozygote, you might be knowing. Recessive, this is homozygote. RR, this is homozygote. This is heterozygote.
Heterozygote.
Okay. So they are expressing, they are operative only in the homozygote condition. So you have to see if they are in the recessive condition or dominant condition. This is dominant. बहुत difficult तो नहीं लग रहा है concept. देख लेना. You can stop me whenever you are getting a heavy bouncer concept, right? This is heterozygote, right? So there is a different effect of the mutation when they are in the recessive. They are autosomal recessive, right? Means they are not related to the sex chromosome. There are two types of chromosome, na? Autosome and sex chromosome. Right? So if they are if the mutation is there in the sex chromosome, then this is called the sex limited, sex influenced, as you can see in our goal, right? Sex linkage, sex limited, sex influenced characters. So all we are talking about the mutation over the sex chromosome. But when we are talking about the AR, we are talking about the autosomal mutations. So how they are affecting at the phenotypic level. So whenever there is a homozygous recessive condition, then only they are going to show their severity. Clear?
Okay.
Right. Severity means they are going to kill that person or affect severely. Otherwise, if the compound heterozygote condition is there, so they can carry the problem, but they are not very severe in that patients. Another example is, so pedigree, by pedigree you can see, that can be asked in the examination that, suppose this is a pedigree. Now, what type of question ही बना देते हैं. What type of disorder is this? Right? So this is AR, This is sex. Sex limited.
Sex linked.
So what you can see, some sex limited I'm writing. Sex linked, sex influenced. So if you are such character, then such question, how will you identify? Then you have to see if the transmission is there in the male and female, both male. If the both male and female is going to be siblings are going to be affected, then certainly this is not the sex related mutation, right?
Yes.
This is not going to be a sex related mutation. And if the siblings are showing the characters, it is certainly
Autosomal recessive.
means you got the point. I can ask 100 questions, but you have to see the black one.
Yes.
Nothing to worry. Automatically, your hand will go here to mark the right answer with confidence. Okay,
Yes.
you can ask different questions that how many child is going to be affected, how many female are going to be affected, how many male are going to be affected. What will be your answer? 50% female will be going to be affected and 50% male is going to be affected in the condition of recessive. Clear? That's you have not to go into the detail. Sometimes this condition is called as the pseudo dominance.
Okay.
Right? Pseudo.
Dominance.
So we are learning a lot of things. बहुत सारी चीज हम सीख रहे हैं. Okay? That is very good. So what you have learned today? Tell me. क्या सीखा भाई?
Phenotypy, pleiotropy, genome silencing,
Ah, yes. A lot
autosomal.
of things we are phenocopies and phenocopies we सीखा.
Yes, sir.
So in very limited time, we have learned a lot, and we have discussed the conditions also. So
Yeah.
that is useful when we are getting the questions from section C or section B. Immediately, without wasting time, you can answer. You can answer easily without any problem.
Okay.
Yes. Hello. Check. Hardy, do you know? Have you any idea about the Hardy-Weinberg rule? Hardy's rule. That is called the Hardy's rule.
Yeah.
So exactly, Hardy's rule is related to the
Homozygous.
population genetics. This is related with the population genetics. Right. So already you might be knowing there is a dominant gene, recessive gene. The dominant gene can be called as the P, right? A recessive gene is called as the Q. And if you are making the heterozygote, they can be called as
2PQ.
okay. So, so that is the formula P plus Q square is equal to 1. Square is equal to 1. This is mathematical expression. So P square plus 2PQ plus
Q
square
square
is equal to 1. So this is, we have to remember this is dominant This is the recessive gene in the population, and this is the heterozygote population. Heterozygotes. So you are going to have a different situation in the population. Gene frequency will be given of recessive or dominant, right? They can ask you the heterozygotes, or they can ask you the dominant frequency or recessive frequency, right?
Yes.
So that you can calculate based on this formula. Always remember this kind of formula, and we will make you practice over that. No issue. We have to discuss. What is the incomplete dominance? What is the feature of the incomplete dominance?
Dominant allele does not dominate completely, sir.
Yes, and always you can identify there is genotype and phenotype ratio is same. 1 to
Yes,
1.
sir. Yes, sir.
Always 1 to 1. Okay. So yesterday we have discussed what the quality square, how the genotype. If you want to make me revise, we can revise also that concept, how we are calculating the genotype and phenotype preferences. Example you have to remember perfectly. This is familial hypercholesterolemia. आजकल ये पूछ रहे CSIR में. These questions are very focused in the examination because if anyone is asking about the incomplete dominance, the students are relating with the rules, right? But what are the
Yes.
animal examples? You cannot remember exactly. This is hypercholesterolemia. Everyone is facing today's time. Right? What is hypercholesterolemia means? Elevated level of
cholesterol.
blood serum cholesterol. And how this is possible? This is only possible because of different type of gene expression. In either case, if the recessive homozygous condition is there, they are inability to make LDL receptors. So that's why this is a result in the high cholesterol level, because there is no receptor to receive them and process them, right? So you can see in this case you are getting a severe disease. But if this is a heterozygous condition, if the genes are there, you are getting the mild disease.
Yes.
But in the perfect dominant homozygous condition, all the cells are normal.
No.
All the cells are all the cells are normal.
Selection లో మనకు
Right?
అప్లైడ్ నేమ్ రావట్లేదు నాట్ సీయింగ్ ద కాంట్రిబ్యూషన్.
So you got the idea, incomplete dominance in case of human being. And if I am not wrong, this question was asked last year about the hypercholesterolemia expressions. And they were not focusing on the genetic, they were totally focusing over the cause of the severe disease. They might be asking the half questions only, right?
Okay.
I'm explaining you the full. What is the reason? Because the receptors were absent. Why the receptors were absent? Because of homozygous condition. And because of homozygous condition, there was no
LDL receptor.
Good evening, sir. बच्चों, sorry, there was a urgent call. I'm audible?
Yes, sir.
बच्चों आवाज आ रही है? दीपांशी?
Yes, sir. Yes, sir.
Yes, Pandit. Am I audible to you? So he left. His expression gone. है ना? तो वेरी इंटरेस्टिंग टॉपिक, मुझे बड़ा इंटरेस्ट आता है इस सब में, बिकॉज अगर बिजनेस पॉइंट आप सोचो तब भी ये काफी हेल्प करता है ना, कि इफेक्ट ऑफ एनवायरनमेंट कैन चेंज एक्सप्रेशन, राइट? जीनोमिक इम्प्रिंटिंग, साइलेंसिंग ऑफ जीन, है ना, एक ही कितना बढ़िया नेचर ने बनाया है कि एक में अब बहुत सारी जीन होती है बट दे आर नॉट एक्सप्रेसिंग एट ऑल, है ना, all of them are not going to express, only few of them is going to express. So if you are going to understand in the conceptual way, na, so all you can teach, because you are going to be future teacher, future researcher, right, professor level, then you have to not, I'm not requesting you to go and do the hard work, rather than I'm emphasizing on what? Understanding the concept, because I'm there to do the rest of the work for you. Just you have to visualize the concept, think the concept, apply the concept, and that's it. That's the reason of most of the students are getting success after getting such kind of lectures. Okay. So another example, very good example, is incomplete dominance. Right. So what is the incomplete dominance in case of Tay-Sachs or sickle cell anemia disease? Can you express this diagram? Can
Yes,
you
sir.
explain this diagram?
Incomplete dominance में जो heterozygous होगा, they are carrier.
Yes, they are normal. Sorry, carrier. Heterozygous condition, they are carrier. And if they are homozygous dominant, they are normal.
Normal.
But when they are expressing the disease, homozygous recessive. In section C, such kind of graph can be given, and they can give you the 4 type of conditions related to the disease, right? And they can ask you which sentence is true, which one is
9015.
sentence. You might be evaluated based on your understanding of this concept. I think now you have a complete understanding of incomplete dominance. First
Dominance.
important thing,
The result and
1-to-1 ratio, genotypic and
Yes.
phenotype.
Phenotype.
How the
Phenotypic and genotypic ratio is
Is same.
equal in
and
incomplete dominance.
yes, why cholesterol is going to rise in the body in some persons?
it should not be
देखो ना, बहुत मोटे होते हैं कुछ लोग ना,
हाँ.
इतना मोटे होते हैं कि उनको क्रेन बुलाना पड़ता है उठाने के लिए, उठ जाओ भाई, कार में नहीं जा सकते, इतने ज्यादा मोटे होते हैं. They are not
location
of some
supplies.
problem, rather than how you can explain why they are so fatty or having so high level of cholesterol.
You can search.
What is the reason?
ఇక్కడ రావట్లేదు సార్ మనం సెర్చ్ పడిన క్లిక్ చేస్తానే
Panchi,
ఇవి రావట్లేదు.
can you
Hypercholesterol.
explain? Brahma Kalyani,
Sir,
ఓకే సార్.
can you explain why the person is getting very high level of cholesterol here?
sir, I'm
Just
not.
figure out one sentence. I'm asking the question in case of hypercholesterolemia, hypercholesterolemia, why the person is getting the severe disease?
Due to
Just a simple
homozygous recessive alleles,
Homozygous recessive
so there is
condition.
no expression of LDL receptors.
Yes, yes, right.
Due to
Very
that, high cholesterol is
Because
incorporated.
the receptor are not going to pass them inside the cell for the further metabolism. So LDL receptor is required, which is not going to be formed whenever they are in the homozygous recessive condition. You got the idea? So you should not forget such kind of concept. And what is the codominance? Codominance.
They are competent to each other, but
They are showing the same level of penetrance whenever they are together. Individually, A is dominant, individually B is dominant, but when both are together, they are expressing equally, right? So
Yes.
that's why you can see they are reacting over anti B. A is reacting over anti B and B is reacting over anti A because the gene is there, IBB, IBB, IO, right? And in the case O, what you can say, there is no co-dominant type of expression, right? So co-dominant you can see only in the case of AB blood group. Only in case of AB blood group, this is dominant. Be analytical, right, beta? Got the point? If I'm saying co-dominance and showing all these diagrams, so you are thinking this is also co-dominant, this is also co-dominant. This is not the case.
Only AB.
This is recessive. This is AB co-dominant, and these two are
dominant.
Got the idea? That's why if a person, if a female blood group A is marrying with the male blood group B, so mostly their children will get the which blood group? AB.
AB.
All right. So B blood group is not advised to marry with the B positive. All right. Because marriage to hoti hai, they are not going to check. Anyone is not going to check today. They are just matching the gotra and all that, right? They should not belong to. No one is going to check, but future chances are there. People will see, they are going to also quickly check out the genetical map, right, of a person before the marriage. They have the dominant gene or recessive gene because no one want to have the cholesterolemia or CFTR, right? So the science will be so advanced that you can predict your child before their
But
before they rise in the world. So you can know everything, every possibility you can check. Which person is going to have recessive gene, or they are inheriting from their parents or grandfathers, right? So complete genetic analysis will be recommended after a few years because population will be so heavy and if the radiation is high and pollution is heavy, then you can see everyone is going to have some kind of disease. That is, tackling is very tough, na? Because anyone is not free. Everyone is making consuming time over different type of aspects. So that's the concept of multiple allele. Why we are talking about the multiple allele? Because there are two type of allele. If you are talking about the blood group A, then you have the two alleles, IA or IAIO. Right? If you are talking about the blood group B, you have two type of IB
and
IB or IO. So that's third type is involved. That's why we are calling them as a controlled by the alleles. And because of the three type of genes basically involved, you are getting the four type of blood types: A, AB,
B, O.
A, B, O. Right?
Yes.
Here. And when you are getting the blood group O, and both the recessive condition, right?
Yes, sir.
Yes.
Both the recessive genes are there. If anyone is there which is dominant, in this case, B blood group. In this case, A blood group.
A.
In this case, A blood group.
A.
In this case, A blood group. In this case, AB. So
B.
how many AB are possible? How many
25%.
AB are possible? Only one.
One.
All right. A blood group, 3.
Hmm.
B blood group, 1 2 3 है ना, recessive is 1, A blood group is 4 I think,
4.
Yes.
है ना, this ratio is 4 3
2,
3
3, 3.
1. No,
No,
A
sir.
is 4, AB is 1,
AB is
B
1,
is 3,
B
and O is 1.
1.
Ratio is 4 3 1 1. Right? So this can be a possible question. Just
So
देखो कितना अच्छा डिस्कशन चल रहा है, and I invited so many students. Almost, can you guess how many students have invited today? Almost 1000.
Oh my God.
So this is kind of gene activation. Recessive gene is there, they are not get activated. है ना? If the dominant gene is there, just joking. Okay, really I have invited so many people.
सर ए बी 2 होगा
ये भी 2 कहाँ है?
सर, यहाँ पे जो फर्स्ट में है ना, सेकंड वाला, उसमें ए बी है एंड ए आई ए है, सो ये ए नहीं होगा शायद
चेक कर लो प्रॉपरली.
सर, ये वाला.
हाँ सही करेंगे, यहाँ पे होगा IP. हाँ ये 2 होगा, करेक्ट. ठीक है. ओके, कैन वी कंटिन्यू?
यस सर.
अच्छा लगा आज की क्लास? कुछ इंटरेक्टिव एक्सरसाइजेज हुई है ना? कोई ब्रेन पे ज्यादा है ना? so many learnings we did today without making so hard effort, है ना? You have not to, you shouldn't make a note, but this is not such kind of concept that you cannot memorize it. You can memorize it. Of course you can memorize it. And then next topic is penetration and expressivity. Therefore, there is a good news. There is one student that qualified JAM, IIT JAM. Do you know the IIT JAM? Have any idea about the IT Jam?
No, sir.
IT Jam is related to the MSc, MTech kind of BTech entrance, the different IITs. Okay. So BTech MTech admission test, you can say. So one student was got jailed by the Bihar government, and then from the jail he started preparing, and now he's in the top list, and he got the 54 rank all India in the IT Jam. So
Wow.
that's what you can say without any teacher, without any books. Right? So anyhow, he managed to arrange some books, and he struggled a lot of with depressions, a lot of emotion you can understand that is in the jail. And he got the 54 rank. So what you can say that your will, powerful will can make you successful. If you are confident, if you can trust yourself, trusting yourself is the biggest motivational point. Trust yourself that you can win, you can understand, you can crack the exam. There is a no tough thing. Every day you keep on revising this kind of things. So today we are not going to, I think mostly we have discussed. Epistasis we will discuss tomorrow. Okay.
Okay.
So tomorrow means Monday, because tomorrow is off, Saturday is off mostly. Monday to Friday we are working.
Okay.
So I
Okay,
hope
sir.
you have liked the class, and
Yes,
every
sir.
day you will try to join and gain some knowledge. And also I'm confident that you can crack the examination if you are regular learning, applying the concept, right? Because this is directly going into the mind. That the penetrance of my lecture is going to be 100% if you are attending the class daily. Okay, I'm sure. Okay.
Yes.
Thank you so much. Good night.
Thank you, sir. Good
Be
night.
positive. Always be positive. Good night.
Good night.